Welcome back, everyone. We had just a few minutes there for a break, so hopefully everyone got a drink of water and is ready for the next talk with Travere Therapeutics on patient-centric drug discovery. I'd like to introduce Shika Denyo, the Associate Director of Patient Advocacy, and also Carson Baumgater, Associate Director of Translational Biology there at Travere. Thank you so much for being here, both of you. We definitely treasure our partnership with you and the relationship that we've built. So go ahead and take it away. Thank you.
Thanks, Roberta. I'm going to kick us off. Good afternoon, everyone. I'm going to start by congratulating Cher, Roberta, and everyone involved in putting on this fabulous symposium. Once again, you've outdone yourself, and I want to congratulate Roberta for pronouncing my last name correctly. Well done. So this morning we were presented with a series of fantastic talks diving into the science. I was blown away by the progress that Emma, Stacy, and some of the other speakers have made in the past 2 years. And I'd like to apologize to the scientists in the audience because I'm not going to follow suit today. Since this is the first chance for me to present at an Alagille Symposium, I instead wanted to spend this time to introduce Travere Therapeutics and to familiarize you with our mission and our culture. So yeah, my name is Carson Baumgater, and I've been working at Travere Therapeutics for a little over 3 years. As you may be able to deduce from my last name or from my accent, I'm originally from Germany, and I came to the US after completing my PhD almost 15 years ago to continue my training in academia. So this is research at universities or in public research institutes. And after 5 years there, I became a little disillusioned with the mission. And my experience was mostly around publishing new findings before someone else does, and to make sure that you got credit for it. So I decided to switch to drug discovery, initially in a startup company that was targeting was developing treatments for large diseases such as Alzheimer's and stroke. And it wasn't until I joined Travere that I felt like I finally found what I was looking for. It was the very reason why I started out on this career track in the first place, and that is to try to make a difference by helping people. On a side note, I have a side note here that I made for David. I want to thank him for reassuring me that my transitioning from brain research to liver research actually constituted a step up in the ranking of the most important organs in the body. So thank you, David. Anyway, at the time, Travere was still called Retrophin. I will give you a quick insight into why we decided to rebrand in just a few minutes. Okay, I'm not moving the slides. Just give me a second. Here we go. So approximately 4 years ago, Retrophin and the National Center for Advancing Translational Sciences, or NCATS, were approached by the management of ALGSA. NCATS and Retrophin had previously embarked on a research collaboration that included a patient advocacy group as an equal partner. And the message from the leadership of ALGSA was basically, we want what they have. Both Retrophin and NCATS immediately jumped on the opportunity to work with Alagille Alliance. And this three-way collaboration was born and kicked off in the summer of 2019. Our goal is to develop a therapeutic specially tailored for patients with Alagille syndrome. In this endeavor, Roberta and Cher ensure that your voice is heard and is able to direct us in our efforts. We greatly benefit from the patient perspective and the experience you have, and we continuously reminding us of our mission, that is to help patients in any way we can. I think importantly, we are equal partners in this endeavor. As this collaboration suggests, a critical maxim for us at Travere Therapeutics is to work together. And we feel so strongly about this that we decided to rebrand with a name that more aptly captured this. So late last year, our new name, Travere Therapeutics, was revealed and replaced Retrophin. So I want to give you a little bit of background on the name. It is inspired by the Latin for path, which is tractus, and for truth, which is there. After almost a decade in rare disease, we understand that the path with a rare disease can be a windy one, shown in our logo. It's our commitment to walk it with the patients that we serve. Each triangle in this mosaic, let me see, this mosaic here, is a person or a point of support, maybe a family member, a caregiver, friend, an advocate, or a healthcare professional. And we see ourselves among this community, coming together with others to support patients on their journey. The different colors of this mosaic represent the diversity of our team and the unique experience, knowledge, and perspectives. It also represents the many stakeholders in the rare disease community. Now, we often add the slogan, "In Rare for Life," under our logo. And that is a statement about our commitment, but also the vision that we have, and that is to make life equal for everyone. Now, my colleague Shika is going to provide you with examples of how we support and help patients outside of our actual research efforts later on in this presentation. Now, physically, we're a small biotech company of around 200 people based in San Diego. We have a focus on rare liver and kidney disease. So this slide shows our drug pipeline, which includes both our approved therapies and those that we're still working on. I want to draw your attention to these two in particular, Colbaum and Kinodal, which are bile acid replacement therapies that are prescribed for rare liver diseases. Diseases like Alagille syndrome, which in the majority of patients affects liver function, but is of course not restricted to the liver. I want to share this with you to highlight that we are a company that has experience and a footprint in rare liver disease, and that we understand the challenges that come with it, the unique challenges that come with it. Now, I know this is somewhat a platitude, but we really live by this. Collaboration is the key to success. We try as much as we possibly can to work with physicians and researchers at centers of expertise, such as CHOP, Lucille Packard, and Cincinnati Children's Hospital. We rely on people with invaluable experience in the field, such as Nancy and Kathy, to vet our ideas and our research. We also work with companies where it makes sense, with other companies where it makes sense. So for instance, Travere and Travere decided earlier this year to co-promote Advocacy, which you just heard about, because we have a network of pediatric hepatologists at Travere that was formed through our efforts with the bile acid replacement therapies. So this means that once Advocacy is approved, Travere can get their drug to more patients that need it faster. As part of our project, we are developing new tools and making them available to the research community. For instance, we're generating valuable disease models that we publish about and that can be obtained and used by other scientists to advance research into Alagille syndrome. Now, it's taken Miriam and Albereo many years and a lot of hard work to get to where they are now. Bringing a drug to the market is no small feat, and kudos and congratulations to both of them. But I want to be clear, we are still early on in our campaign. Our efforts are rooted in the conviction that more treatment options translate to a striking benefit to patients. Not every patient responds equally well to a given drug. Options are an important way to ensure that each patient is able to access a therapy that helps him or her. Also, in many cases, treatments are complementary and can be used jointly or alternately for better outcomes. So we're approaching the challenge of developing a new, So how are we approaching this challenge of developing a new therapeutic for Alagille syndrome? Well, I stole this figure on the left here from one of Emma Anderson's papers. It very simply illustrates that JAG1 is a protein on the surface of one cell that signals to notch receptors that are expressed on the surface of another cell, and instructing this latter cell to respond. Response may be different depending on the type of cell that receives the signal and other co-stimulatory signals, but these are not shown here. Now, multiple speakers have already alluded to this, but to support my next few points, I do want to go over it once more. Most Alagille syndrome patients carry mutations in the JAG1 gene and a small percentage in the NOTCH2 gene. Importantly, each person carries 2 copies of each of these genes, and in patients, only one of these bears a mutation, while the second one is intact and fully functional. As a result, there is less signaling via this axis, and this results in the disease hallmarks that are characteristic of Alagille syndrome. David introduced therapeutic concepts on a high level yesterday, and we've already heard from speakers this morning about therapies today, or therapies in development. So I want to give you my perspective on what therapies for Alagille syndrome could potentially look like, and maybe a little bit of a ranking of these. Now, inarguably, the ideal therapy would replace or correct the mutated JAG1 or NOTCH2 in a perfect manner. When I say that, I mean that the product of the gene, that's the JAG1 or the NOTCH2 protein, would be produced in the right tissues, in the right amount, and at the right time. Now, with our current technology, that is extremely difficult, or maybe even impossible. But this technology is evolving rapidly, and I think that this may change in the future. The second best option would be to upregulate the intact copy that is still functional. Now, we would expect beneficial effects if we were even able to increase the JAG1 protein signal from 50 to 70%. And that's just a rough number. I'm just making a point that a slight increase in the amount of JAG1 signaling may have a beneficial outcome. Now, thirdly, we could replace the biochemical function of these proteins. For instance, if we knew that another member of the JAG1 protein family, a sibling or cousin of JAG1, could fulfill the same function, then we could try to activate that protein. Another example would be to increase the gain on the response to the Notch receptor, so that a reduced JAG1 signal may be offset. And I believe Du Dong is going to talk about this later on this afternoon. A fourth approach would be to compensate for the loss of biological function of JAG1 Notch signaling. In this case, a completely independent signal that has the same effect in cells may be able to make up for the deficiency in JAG1 Notch signaling. And a final approach I want to highlight is to treat individual symptoms of the disease and thereby provide relief to patients. Now, this list is by no means comprehensive, but I want to give you a glimpse of the type of therapies we can conceivably try to develop. Just because it's conceivable does not make it realistic, and it is really up to us in the drug discovery campaign to identify those that have potential and those that do not. Okay, so I wanted to really finish by introducing you to the core team at Travere and NCATS. On the left here, we have our NCATS collaborators. We have Ivan and Philip, who are postdocs at NCATS. We have Atina, who is the project manager, and Wei, who is our scientific project lead at NCATS. Then we have Steve Rodems, who is our head of research at Travere, and myself, and I'm a scientific lead on the Travere side. Now, our extended team includes other members of our organizations, as well as consultants and, of course, service providers. I think most importantly, however, it includes all of you. With that, it's my great pleasure to hand over the microphone to my esteemed colleague, Shika Dunya.
Thank you, Carsten. And my spin cycle is going. So if you hear noise, sorry. So as Carsten just stated, my name is Shika Dunyo. I'm the Associate Director of Patient Advocacy at Travere Therapeutics. I started with Travere in March of 2020, so right in the middle of the pandemic. Prior to joining NORD, I was the Director of Education Programs, where I was responsible for developing educational resources for patients and caregivers, medical professionals, and students. I'm based in Washington, D.C., and at Travere, we talk a lot about our why we do what we do, why we work in the role we work, why we work at Travere. My why is my sister. and my sister passed away from a rare autoimmune disease in 2008. So I consider myself a rare disease advocate and a rare disease caregiver. In my role at Travere, we in patient advocacy like to say that we have the best jobs in the company. My job is to collaborate with patient advocacy groups like Alagille Alliance to ensure that we're providing the right kind of support for the communities that we serve, and that we're bringing in patient and caregiver insights into the company to ensure that we're patient-centric. Next slide, Carson.
Sorry, hang on. Here we go.
Thank you. So in that my focus is to ensure that we are providing the right kind of support. And so this year, we're proud to be a sponsor to many of the Alagille Alliance's programs and initiatives that provide education, access, support, and financial assistance to the community. These programs include this year's conference, the financial assistance program, the bereavement and support group programs. These programs really are important in alleviating the burden that many families feel as they face the challenges that they're walking through in their journey. Next slide. Carson, next slide. Oh, thank you. In addition to that, we also want to make sure that we're being guided by the lived experiences of the people that we serve. And so this year we developed a new patient advocacy council. It's an advisory group made up of patients and caregivers. And our goal in developing this council is to make sure that we are hearing from patients and caregivers, that we're gathering their feedback on patient materials, on clinical trial design, and any other programs and initiatives that we are developing. I'm proud to say that 2 members of the Alagille community, one of which I believe is still online right now, are members of the council, and they've been really engaged thus far and have provided great insights to our young council. Next slide. In addition to supporting and gathering insights, we want to make sure that we're also educating the community. And so we have provided support to NORD for the development of an educational video on Alagille syndrome. And recently, we provided additional funding for that video to be translated into Spanish so that it can reach many more people within the community. What's great about these videos is, and because I actually worked on this initiative, I know these details, but these videos are developed in collaboration with a company called Osmosis. And Osmosis has a platform, a subscription platform that is available to medical schools around the world. And so these videos are not only accessible through NORD's website, it's accessible to medical schools around the world. And so it's wonderful that these educational videos, including the one on Alagille, are gonna educate budding physicians. And so it's such an amazing initiative, and I'm glad that we have funded videos for Alagille. Next slide. And then finally, in addition to support, gathering insights, providing education, we also want to make sure that we are raising awareness, both internally and externally. And so another initiative that I'm proud of is our Rare Life initiative, where we highlight stories from the communities that we're serving. And so we highlight these stories on our website so that people more broadly can see these stories. We also circulate these stories internally on our intranet and via our email. And so earlier this year, we featured baby Delilah and her family And then later in the year, we actually invited Delilah's mother to provide a guest blog on Mother's Day, which was such a beautiful reflection that she wrote about her role as being a mother to a daughter with Alagille. I would encourage you all to take a look. I believe we have it linked directly from our booth. So please take a look at that. And so before I close, I would say I would be remiss, and Carson already sort of called to it as well. All of this is in collaboration with Cher and Roberta. I would not be able to do any of these things without their collaboration and their partnership. It's been a pleasure working with the two of them. They are fierce advocates for the Alagille community, and I'm constantly inspired by their commitment. And so I thank them both for their hard work constantly. And so next slide. I want to thank you all for listening, and our information is here, Carson and my email addresses. So if you have any questions, feel free to reach out. Otherwise, I'm happy to take any questions.
Thank you so much, you guys. Really appreciate you and all that you do. We definitely appreciate our partnership. Any questions?
Did you see my clapping hands, Shika?
Oh,
when
no.
You were talking about the video being translated into Spanish.
Yeah, and just in time for this conference. I'm so excited, and hopefully I look forward to hearing feedback from your community, especially from your community, your Spanish-speaking communities, on how effective it was, and you know, if it's brought value.
Yeah, definitely.
All right, well, the next session is at 3:30. So if there are no questions, we can hop out of here and everybody come back at 3:30 to the next one, the discussion with Mirum Pharmaceuticals. Thank you, guys.
Thanks, Carson. Thanks, Shika.